In this video, we're going to talk about another exception to Mendel's rules that he observed, and that rule is when genes are carried on sex chromosomes instead of autosomes. So another one of Mendel's assumptions is that, again, he didn't really use the word autosomes back then, but that genes are carried on autosomes, which are the chromosomes that carry genes that control for everything except for sex. But as we're going to see today, that's not always the case. And as a result of that, you might see some interesting ratios as outcomes. The last video included when degrees of dominance were not complete and when you had more than 2 alleles. In this video, we're going to talk about genes on sex chromosomes. So just a little reminder again that genes are found on chromosomes, and you inherit the chromosomes from your parents through their gametes, through the sperm and the egg. And then based on the collection of chromosomes you get a certain collection of genes, and then that determines your characteristics. And Mendel didn't call them genes back then, he called them hereditary units. Now I want you to get the idea that gametes are very diverse based on the processes that occur within meiosis. And again, based on the gametes that you get, you'll have different characteristics. So since there are such a large variety of gametes, you can get such a large diversity of individuals within populations. And the reason different chromosomes leads to different groups of chromosomes leads to so much diversity is because different groups of chromosomes have different genes on them. Another important term to remember is the location of a gene on a chromosome. So let's say there's a gene over here. Let's say it's for eye color. I would call that the locus for the eye color gene. So the location of a gene on a chromosome is called the locus. Plural would be loci. So I would have 2 gene loci over here, L-O-C-I. Here we can see some coloring of gene loci, or a gene locus over here. This is on a replicated chromosome, so we know that this gene here, or this gene here, and this gene here would be identical to each other. But the point is that that band shows a location of a gene, so it's called a gene locus. So another little reminder, you have a karyotype over here of chromosomes in a somatic cell. We know that because there are 46 chromosomes. The type of inheritance that we've been studying so far is called autosomal inheritance, where essentially you inherit genes that are carried on autosomes, the chromosomes that are in the first 44 pairs. But autosomal inheritance is not the only type of inheritance that exists. You also inherit another pair of chromosomes called your sex chromosomes, and those sex chromosomes have other genes on them. So whenever you have genes that come from a sex chromosome, we call that sex-linked inheritance, when you have genes on sex chromosomes. For autosomes, both males and females would experience the same pattern of inheritance because it's not linked to the sex. Whereas in sex-linked, you might notice different patterns of inheritance, different ratios in the males and females if a gene is found on a sex chromosome, since it is related to the sex of the individual, boy versus girl. So again, sex-linked inheritance is when you inherit a gene that's found on a sex chromosome, either the X or the Y. If the gene is on the X chromosome, we call it X-linked. If the gene is on the Y chromosome, we call it Y-linked. You should be able to predict if something is a boy or a girl based on their karyotype. So over here you can see we have 2 X's, that would be a girl. And any gene found on the X chromosomes would be considered an X-linked gene. Remember that boys will get an XY pair and girls will get an XX pair. And it's that small difference that makes the boy a boy. So having that Y chromosome will make the boy a boy, and having 2 X's will make the individual a girl. Remember that the X will always come from, the X in a boy will come from mom, and the Y will always come from dad. In a girl, 1 X will come from mom and 1 X will come from dad. So the dad is really determining the sex of the individual based on what he gives. If he gives an X, it's a daughter. If he gives a Y, it's usually a boy. Just a quick reminder, remember homologous chromosomes, you get one from dad, one from mom, same length, same gene location, can have different version of genes. Those different versions of genes, I wasn't using the word when I first introduced this, but they're called alleles. So one chromosome might have the big A version, the other chromosome might have the little a version. It's important to remember the homologous chromosomes because your sex chromosomes are also homologous chromosomes, but we sometimes call one particular pair, the X and Y, the odd couple of homologous chromosomes because they're not quite similar in every way. They don't necessarily have the same genes everywhere. X has a lot more genes than the Y chromosome. They don't have the same length. They don't have the same centromere position. They do have some regions of similarity. They have to if they're to pair up during meiosis, but they're kind of the odd couple. So like I said, the X and the Y are the odd couple, but they do have some homologous regions so that they can pair up during meiosis. If they didn't have those homologous regions, they wouldn't be able to pair up. So this over here, this yellow and yellow, that would be considered the homologous region. Now this is not in a human. How do you know this is not in a human? Well, because the Y is longer than the X. So this over here is the XY system in a human. So we have our Y over here, which is shorter, and our X over here, which is longer. Homo sapien, just the name of of humans, the scientific name for humans. The pairing region is a region where they are similar to each other. So on the Y chromosome, there's a gene called the SORY gene, SRY gene, and it's that gene that triggers development of male characteristics. So usually what will happen is the baby will start to develop, and by default it will have characteristics that look more female. So for example, it'll have let's say nipples. But now if you have the SORY gene, those nipples would not develop into breast because the SORY gene will eventually trigger male development, and then that child would become a male individual later on. So we can see the SORY gene over here. It would trigger a cascade of events that would lead to male development instead of female development. So you can see the idea here. This is before sex is determined. If the SORY gene is present, we're going to go to this route over here and become male. If the SORY gene is not present, we'll go this route here and become female. So we can see here a zygote is made. Over here, we don't know what the sex of the individual is going to be yet. After about 2 months have gone by, the SORY gene will become active if it's present. If it's active, testicles will develop. That'll make more hormones and lead to the male phenotype. If the SORY gene is not present, then you'll have the development of ovaries, female hormones, and therefore the female phenotype. So if you're really interested in finding out again in some more detail why men have nipples, even though we don't use them, you can watch this link here, and there's a great book that you can read that discusses the whole concept in addition to a bunch of other interesting scientific questions that you might have. So, as I mentioned before, any gene located on a sex chromosome is called a sex-linked gene. Any gene on an X chromosome is called an X-linked gene specifically. We're going to see later on some experiments that allowed us to discover sex-linked genes. They were done by a scientist named Morgan. He experimented with fruit flies, and we're going to see the reasons why he selected fruit flies. But in summary, the major ideas that he came up with is that fathers pass on X-linked genes to their daughters, but not their sons, because they give Y to the son. Mothers are able to pass on the X-linked genes to both boys and girls. So father can't pass on X to son, but they can pass it on to the daughters. And then the mothers can pass it on to both, which is a big key takeaway from the experiments that Morgan had conducted. So again, if you have a gene on a Y chromosome, that's called a Y-linked gene. Hopefully you'll understand that only males can really exhibit a Y-linked gene phenotype because only males have the Y chromosome. And hopefully you also understand that there's more X-linked genes out there because the X chromosome is longer than that tiny Y chromosome that males have. So if you have Y-linked genes, so this picture here kind of shows you who can pass on a Y-linked gene. The individual who's colored in pink over here has a mutation on the Y chromosome that led to some kind of disease. And so if you have the disease in this diagram, you'll be colored pink. The mother doesn't have that Y-linked disease because she doesn't have any Y chromosomes, so she's in this gray color here. Now you'll notice they have kids, and the only individuals that are affected are the boys, because the boys are the only ones who can get the Y from the father. The girl doesn't get the Y from the father, she gets the X, and because she gets the X from the father with no disease on it and the X from the mother with no disease on it, the girl will not have the disease. But the sons will be affected by a Y-linked gene. So we're going to take a look at more examples of patterns of inheritance with Y-linked genes and X-linked genes as well. So before we move on to some Punnett squares with X-linked and Y-linked genes, I just want to take a look at what are the chances of getting a boy versus getting a girl. So remember that the child will always receive an X from mom, so you always get an X from mom no matter what. So it's a 100% chance of getting an X from mom. The only real deciding factor again is what you get from dad. And so if you get Y from dad, you're a boy. If you get X from dad, you're a girl. And it's very close to a 50-50 chance. The father could have a 50% chance of giving you a boy, 50% chance of giving you a girl. Remember that it's a probability though, it's not a guarantee. So if you have a family with 2 boys, it doesn't mean you were ripped off by Mother Nature. It just means that it was a probability all up to chance. Now, in reality, there actually seems to be more boys than girls in population, so it might not actually be 50-50 like we think. It might be a little bit higher for boys than it is for girls. And so there's a neat article here you can read to find out more about it. One interesting theory is that sperm that contain the Y chromosome have a smaller chromosome than sperm that contain the X chromosome. And because this chromosome, the Y chromosome, is smaller than the X chromosome, these sperm are just faster and then reach the egg a little bit faster, and therefore they fertilize more eggs. That's just one kind of going hypothesis behind the whole process, but there's more discussed in this article if you're interested in finding out more. For the purpose of the course, though, we'll just assume that it's roughly 50-50 chance. So we're going to talk about some traits called recessive X-linked traits. Recessive X-linked traits are traits that are, again, not dominant. They're recessive, so they can be masked by a dominant allele, but they're found on the X chromosome. So, for example, if there's a recessive X-linked trait and a female is X_r, that female will have the recessive trait. Now, if the female is X_r, X_r, the female will just have the dominant trait instead of the recessive one, since this dominant takes over. So when we represent X-linked genes, what we do is we use X as our base letter and then a superscript as whether you have dominant or recessive for the characteristic. So this female would have 2 X chromosomes. She has the 2 little r alleles. This female here would have 2 X chromosomes. She has a big R allele and a little r allele. Another female might have 2 big R alleles. Just like that. So males, on the other hand, before we go into males, over here, just very quickly, this individual would be homozygous recessive for the little r allele. This individual would be a heterozygous individual, and this individual here would be a homozygous dominant. Now, males, we don't really use the term heterozygous or homozygous or homozygous dominant or homozygous recessive. What we use is hemizygous, because for X-linked traits, males only receive 1 allele for an X-linked trait, because the other sex chromosome they have is a Y chromosome. So for a male, for example, you could have X big R Y. You could have X little ry. So we refer to the male as hemizygous because they only get half of the alleles that you would get on an X-linked chromosome, since they have Y chromosomes, not X chromosomes, as your other chromosome. Now, what you'll also notice is that this boy here has the disease. If the disease is recessive, the boy has it automatically. As soon as the boy gets the X with the little r on it, he has a disease. Now, a girl, on the other hand, for her to get the disease, she needs to get 2 X's with the little r's on it. So there's a smaller chance that the girl gets this disease than the boy, because the boy just needs to get 1. The girl, if she has big R, that will protect her from the disease. So I want you to get that there are different patterns of inheritance with boys and girls, because girls just get more X chromosomes than boys do. So if we're still talking about an X-linked disease that's recessive, if you have a heterozygous girl, X big R, X little r, that heterozygous girl is called a carrier for the disease. So she doesn't have the disease because she has a dominant healthy allele, but she carries the allele for the disease, and so she can pass that on to her kids. Now, males, they can't be carriers for X-linked diseases. And I think you understand why now. That's because males will have X little ry. As soon as they have an X little r, they have the disease. They don't have another X chromosome to mask the disease. So let's take a look at how we came up with the idea that genes can be found on sex chromosomes. So there was a scientist named Morgan. He worked with Drosophila, which is a scientific name for fruit fly. The reason he worked with Drosophila is because they reproduce very quickly. They make a lot of children, so you have a large sample size, and you can take lots of notes and get averages and notice patterns. And they're really easy to study because they don't have a lot of chromosomes. They only have 3 pairs of autosomes and then 1 pair of sex chromosomes, either the XX or the XY. So here we can see fruit flies, and you can see there's a lot of phenotypic things that you can study. You can look at the structure of the wings, you can look at maybe the antenna, you can look at the eye color as well. Some fruit flies will have red eyes, some fruit flies will have white eyes, and so there's many things that you can study in fruit flies, kind of like you could study in pea plants. So in the first experiment that Morgan did, he took a red-eyed female and crossed it with a white-eyed male, and he noticed that you only had red-eyed individuals produced. So from that, he was able to figure out that the white-eye trait disappeared and the red eye persisted, so that means that the red eye trait is dominant. So this is the cross he did. Now, he didn't know they were on sex chromosomes at the time, but I'm going to do the Punnett square as if they were on sex chromosomes. So I'm going to put the red eye individual on top, the pure reading red eye female on top, so X big R, X big R. And then I'm going to put X little r, Y. And then I'll get, to my fertilization event, I'll get X big R, X little r, X big R, Y, X big R, Y, and then X big R, X little r. So you can see we have red, the big R everywhere. So all are red, 100% red. And you can also see you have 50% female. 2 XX individuals, 2 XY individuals. So we have 50% female, 50% male, but they're all red-eye individuals. Now at this point, we couldn't really tell if the eye color is on the X chromosome or on an autosome. So Morgan then decided to take the F1 individuals and cross them with each other. So he took a red-eye female and a red-eye male, and he crossed them together. And he expected the 3-to-1 ratio. So let's see if that happens. So he did an X big R with an X little r, female, red eye from the F1 generation, took a red eye from the F1 generation, male, and he got X big R, so we got a red female, he got X big R little r, so a red female again. He got an X big R Y, so a red male. And he got an X little r Y, a white male, a white-eyed male. And so, yeah, you get your 3-to-1 ratio. You get 3 red to 1 white. But there was a weird thing that happened. For some reason, when he did this cross, it was only the male that would have white eyes. So in this heterozygous cross where you had a red eye female and a hemizygous red eye male, you'd get your 3-to-1 ratio, great, but only the males had white eyes. So that led to the conclusion that eye color is probably carried on a sex chromosome because there's a difference in the sex patterns here. Why is it only males getting white eyes? And he then concluded that the gene for eye color must be on the X chromosome, not the Y chromosome. So the father has red eyes, and the mother has red eyes, but has an allele for white eyes. The mother donated X little r to her girl, but the father gave X big R to the girl. So that's why the girl ended up being red. The mother gave X little r to the boy, and the father gave Y to the boy. So from that we can tell that the X little r must have come from the mother. It was hidden in the mother. And the mother doesn't have a Y. She has an X. So from this experiment, Morgan was able to tell that the eye color gene came from the X chromosome, not the Y chromosome. And so whenever you see interesting patterns in terms of a characteristic and it might be related to the sex of the individual, then you can probably conclude that it's linked to a sex chromosome. If the mother can pass on the characteristic to her sons, then you know that it's an X chromosome linked gene because only mothers give X to their sons. So we'll do a few examples here together. So let's analyze this cross here. So we had X big R, X little r. And we're going to cross it with the X big R Y. And same idea, you do your Punnett square, so we have X big R, X big R, X big R, X little r, X big R Y, X little r Y. And then we have the percentage of, then we're asked a series of questions. So let's take a look at the percentage of offspring that are male. Well, of the 4 here, there's 2 out of 4 that are male, so that's 50% or 1/2. Let's look at the percentage of female offspring. Well, again, 2 out of 4 are XX, so that's 50% or 1/2. And let's take a look at the percentage of red eye offspring. So we have red over here, red over here, red over here. That's 3 out of 4. 75% of the offspring have red eyes. That's out of all the offspring. White eye offspring, just 1 over here. That's 1 out of 4, so 25% chance of having a white eye offspring, but it's always the males. Percentage of males that are red-eyed. So now pay attention to that question here carefully. percentage of males, not percentage of offspring. So we're looking at just the males only here. There's 2 males. Of the 2 males, how many are red-eye? 1. So 50% of the males, 1 out of 2, 50% of the males have red eyes. Percentage of males that have white eyes. Well, out of only the males, that's what we're asking here. Out of only the males, how many have white eyes? 1 out of 2, so that's 50%. Percentage of females that have red eyes. So let's just look at the females. They both have red eyes. 100% of females have red eyes, and 0% have white eyes. So you need to read the questions carefully, especially when you're studying genetics and probabilities. And again, the genotypes will have a base letter. The base letter is which chromosome the gene is on, and then a superscript to show if it's the dominant or recessive allele for that trait on that particular chromosome. And the gametes work in the same way. X big R, X little r can make X big R or X little r. The male can make X big R here or Y. So here's another example of a sex-linked disorder. So we'll go through it together. You can try it yourself, see if you have the right answers as I go through it. So muscular dystrophy, we'll sometimes call it just MD, is a recessive sex-linked disease. It's an X-linked disease specifically, and it's recessive. Write the genotype for a male with muscular dystrophy. So it's recessive. That means you have to have X little a. The male doesn't have another X, the male has a Y. A female with muscular dystrophy would have to have both recessive alleles, X little a, X little a. A carrier of the disease that is female, well, that means that the female is healthy, but carries the diseased allele, so X big A, X little a. A healthy male would have to have the dominant allele, X big A, and then Y. So you should be able to practice genotype writing based on phenotypes, based on what type of sex-linked characteristic you have. So we'll do a few sample problems together here. A male with muscular dystrophy has a child with a carrier. Using a Punnett square, determine the chances of that the couple have a boy, a girl, a diseased boy, a diseased girl, and who gave the disease to the girl, who gave the disease to the boy. So let's write down our cross. So we have somebody with muscular dystrophy, a male with muscular dystrophy, X little a, Y, and it's crossed with a carrier, a female carrier, so X big A, X little a. And so let's do our Punnett square. We'll put the female over here, the gametes for the female, so X big A, X little a. And then we'll put our carrier over here, or our male over here, X little a, Y. And then we'll carry out the fertilization event. So we have X big A, X little a, X little a, X little a, X big A, Y, and then X little a, Y. So what are the chances that the couple have a boy? Well, we can see that there's 2 out of 4 that are boys, so 50% chance of having a boy. And same thing for the girl, 2 out of 4 there that are girls, so 50% chance. A diseased boy. So let's find the diseased boy. So this over here is a diseased boy. So you have 25% chance of having a diseased boy because it's 1 out of 4 chances that you have a diseased boy. A diseased girl. So let's find the diseased girl over here, right here. And you have, again, only 1 there out of 4, so 25% chance of having a diseased girl. Now, who gave the disease to the girl? Well, you could say that both mom and dad did. The mom gave the disease to the girl over here, and the dad gave the disease to the girl over here, so that's a combination there. So both parents did. Now, who gave the disease to the boy? Well, it's an X-linked disease. So the father gave a Y, and the mother gave the X with the disease on it. So the mother gave the diseased allele to the boy in this case. Let's try another problem here. So a healthy male has a child with a diseased mother. Using a Punnett square, determine the chances the couple have a boy, a girl, a diseased boy, a diseased girl, and then we can answer E and F. So let's write down our cross. We have a healthy male, X big A Y, and muscular dystrophy again is a recessive disorder, so to be healthy there better be that dominant allele there on the X chromosome. And then we have a cross with a mother with the disease, so X little a, X little a, so homozygous recessive. Let's make our Punnett square, put the mother on top here, X little a, X little a. Put the father on the side here, X big A, Y. And then carry out your Punnett square, so we have X big A, X little a, X big A, X little a, X little a, Y, and X little a, Y. So let's see, percent chance of having a boy, 2 out of 4, so 50% chance. Chance of having a girl, 2 out of 4, so 50% chance, or you can say 1 out of 2, which is a simplified fraction. A diseased boy, 2 out of the 4 individuals are diseased boys, so 50% chance. Now let's say you do have a boy. Let's say the boy comes out, you see it's a boy, and you know what your genotypes are as parents, then it's 100% chance that your boy has that disease. Because you know it's a boy, and if you do the Punnett square, that boy has to have the disease. A diseased girl, well, 0% chance. There's no diseased girls in this Punnett square. Who gave the disease to the boy? It looks like on both cases it was the mother. Why did the girl not get the disease even though she received the allele for the disease? Well, take a look carefully. The mother gave the girl the allele for the disease. But each time the father gave her an X with a dominant healthy allele. So the girl, because she inherits 2 X chromosomes, one from mom, one from dad, she has a chance of getting a healthy X chromosome. If the boy gets an X chromosome from mom, I mean he always does, he doesn't have a chance of getting a healthy X chromosome from dad because he gets a Y no matter what. So the father protected the girl by giving her the dominant healthy allele in his X chromosome. So that's why it's more likely for a boy to get these X-linked disorders than it is for a girl, because boys do not have the protective factor of getting 2 X chromosomes and getting the chance of being healthy. So the father gave the dominant healthy allele to the girls and only gave the Y to the boys, and they didn't have the chance of being protected. You can try this practice problem out here as well. So pause the video, try it out, and then unpause it and see if you're on the right track. So we have a healthy male, has a child with a heterozygous mother. Using a Punnett square, determine the chances of the following. So let's do our healthy male over here. We have X big A, Y. Let's do our healthy our heterozygous mother. That means she's a carrier. X big A, X little a. We'll put the mother on top over here. So X big A, X little a. We'll put the father on the side over here. X big A. And then Y here. Let's do our fertilization events. So X big A, X big A. X big A, X little a, X big A Y, X little a Y. So again, 1 out of 2 chances for boy, 1 out of 2 chances for girl. What are the chances of getting a diseased boy? Well, you have a 25% chance of getting a diseased boy, or 1 out of 4 chances. If you know that you have a boy, then you don't know yet if it has a disease chance, it's a 50% chance. If you're only looking at the boys, then it would be a 50% chance of having a boy with a disease. A diseased girl, well, there's no girl with the disease here, so it's a 0% chance. There's no chance of getting a girl with the disease. So who gave the disease to the boy? Well, again, it's an X-linked disease, so it was mom. And why did the girl not get the disease again? She got the allele, but the thing is, the father gave her the healthy X allele to protect her from the disease one that her mother gave her over here. So again, the daughter has another protective allele there, whereas the son did not get this chance. As soon as he inherits that diseased X from mom, father can't protect him. Father gives him the Y. So again, an illustration of why X-linked traits, and especially diseases, are more common in boys than in girls in populations. Here are some more practice problems you can attempt. So make sure you go through this, compare answers with your peers to see if you're on the right path. There's some more practice problems over here to try out. These practice problems over here have the answers included, so when you do them you can show your work and then check your answer next to the just below. Here we have red-green color blindness, and this is a test for red-green color blindness. So in this circle, if you can see the number 70 in this little diagram here, then it means you do not have red-green color blindness. So it's type of color blindness where you can't really differentiate the red and the greens. So if you can see 70 in the circle, then you do not have red-green color blindness and you're fine. It is an X-linked recessive trait. So to get it for males, you just need that one X little r. For females, you need both of those X little r's. So you need to get it twice. So females have to get two X little r's to have the disease, whereas males only have to get one X little r to have the disease. So that would mean that males are more likely to get this color blindness disease. And if you don't see that 70 in there, it means you do have that red-green color blindness. And you can see here that 4 to 8% of populations of individuals that have this disease are males. So 4 to 8% of males have this disease, I should say, and then only 0.4% of the females are affected, which supports that idea that males only need one of the diseased allele, whereas females need both, because they have a chance of getting that protective, healthy from, let's say, father, and not get sick. Now, if you thought you had red-green color blindness, don't worry. That was just a joke. You can see the animation here. Now you should be able to see the number within the diagram, the 70 there, and that means you don't have red-green color blindness. So here we can see a summary of the different types of crosses that you might see with males and females, with the colorblind example. So you can see if you have a colorblind father, or a colorblind father, that he'll pass on those colorblind alleles to the daughters, and if you cross it with a normal mother that's homozygous, she'll protect the daughters in those cases there. The sons won't get the disease because the Y chromosome does not carry it, and the father does not pass on the X chromosome to the sons. Now if you look over here, we have a healthy father and a carrier mother. You'll notice that the only individual to get the disease is going to be the son. The mother can only pass on X-linked traits to, or X-linked diseases to her recessive ones to her son. She'll pass on X's to her daughters too, but the recessive ones will only show up in her son because the son will not get a protective factor from the father. The daughters, they will get the protective X big N from the father, but the sons won't. They will get the Y. And then over here we see a situation where the father is colorblind and the mother is a carrier. And in this case, you can get both boy and girl having the disorder because the mother passes on a little n, so does the father, and we have a colorblind daughter. Over here the mother passes on the little n, but the boy does not get a chance to get the protective factor from the father, so he would be colorblind there as well. Here's a great practice problem to try. You have a colorblind woman married to a man who had normal color vision. What would be the probability of what would be the probable phenotypes of their children. So do a Punnett square and come up with those phenotypes based on the Punnett squares that we've seen in these diagrams over here. There are some more great practical problems for you to attempt. The answers can be found at this link over here, and the questions are over here, along with a little description of the disorders and how to interpret the questions above. So make sure you try these, carry them out, and compare with each other to make sure you're on the right path. The readings for this section can be found on pages 200 to 201, and you should attempt the following questions on page 201 and do any extra worksheets and problems within the PowerPoints to test your understanding and to become better at solving Punnett square problems with sex-linked characteristics. And again, this goes against the general pattern that we observe on autosomes, where we see a different pattern in males and females because the gene is carried on a sex chromosome, which is related to the sex of the individual.